Hello and thank you for joining us on another episode of PlantGENE podcast today. I'm Samatha Gunapati as a research lab supervisor at Washington University in St. Louis. If you are new to the podcast or a PlantGENE, so this PlantGENE is a project centered around enhancement of plant genetic engineering to ensure sustainable supply food, feed and fiber. So in each podcast, we welcome esteemed leaders from academia and industry to share their experiences and insights for the benefits of our listeners. So today our guest is Professor Stanton Gelvin. He is a distinguished professor in the Department of Biology at Purdue University. He is also a Ambarga Distinguished Professor Awardee. His research focuses on Agrobacterium biology and also genetic engineering. He is also focusing on understanding the role of some genes and some proteins in this natural genetic engineering to process and how these genes are helpful to improve the agrobacterium transformation efficiency. So today I am delighted to have this opportunity to talk Stan Gelvin, and welcome Stan. Thank you so much joining me today.
Okay, thank you, Samatha. I really appreciate this interview, and I want to thank people at PlantGENE also for recommending that I be interviewed. So thank you very much.
Okay, thank you so much for joining me. And today I'm going to talk a little bit about your experience in science, and I will ask some questions. So can you please briefly describe your current research projects?
Okay, so we have a number of different research projects in our laboratory right now, and they could be fundamentally broken into two groups, basic research projects and more applied research projects. The basic research projects involve understanding how tDNA and virulence effector proteins, once they're introduced into the plant, find their way to the nucleus. We have a lot of data suggesting that the actin cytoskeleton and particular myosin molecules also are involved in trafficking perhaps the tDNA, certainly some virulence effector proteins to the nucleus. So this, and a rather surprising recent result, that VIR-E2 protein, one of these effector proteins, actually seems to localize to plasmodesmata, is a surprise, but these are preliminary data. The second major project that we have in the laboratory is to try to understand tDNA integration. Once tDNA gets into the nucleus, it may integrate. Many copies probably don't, but it may integrate. We're involved in trying to understand two things. First of all, because tDNA seems to be attracted to double-stranded breaks and integrated double-stranded break sites, we're trying to understand how tDNA finds these double-stranded break sites, and then what plant proteins are involved in the integration. Another group recently showed that DNA polymerase theta is important for integration. We also think that DNA polymerase theta is important for integration, but we also think that there are other mechanisms for tDNA integration. So those are two major basic projects. Sort of as a smaller project, we are trying to understand why and how tDNA brings other DNA into the plant cell. We and others have evidence that when tDNA integrates, it could also bring with it DNA from the Agrobacterium chromosome. And we have shown now it could also bring DNA from other plasmids, via helper plasmids or cryptic plasmids. And we're trying to understand how this happens. We think it has to do with recombination of tDNA with these other plasmids or other replicons in Agrobacterium, but we're working on that now. On the more applied, we're doing two different major projects. The first one, Dr. Lanning Li in the laboratory, a research scientist, has now identified and isolated Agrobacterium VIR-D2 mutants that are able to transfer tDNA to plants and reasonably efficiently, but not integrate them very well. And so this has been very useful for transgene-free editing. That's one major project. And another major project is a work that we're doing with my colleague and former graduate student, Kiran Mysore, that we're working together to try to improve use of the type 3 secretion system, introducing and improving the use of type 3 secretion system in Agrobacterium for transferring proteins into plant cells.
Great. You have many projects going on. So I have one more question. So for our listeners who may not be familiar with Agrobacterium, and could you please explain why Agrobacterium is useful tool for bioengineering?
Well, Agrobacterium is useful for several reasons. First of all, it is the only known organism that naturally could transfer DNA between kingdoms of organisms, in this case, a prokaryote and a plant cell. And more importantly than that, Agrobacterium transformation technology is very, very well developed, and it is easy to use in the laboratory and relatively inexpensive also. It does not require specialized equipment the way, for example, particle bombardment does. So it's easy to use, and it's relatively inexpensive, and the technology is very accessible to everybody.
That's great. Yeah. Yeah, it's easy and easy to handle also. I see like a lot of labs, so they do have like biologistic transformations, but these are very expensive method to go through. So, okay, thank you so much. So your research focuses mostly on engineering the Agrobacterium strains and implementing them for transformation. But some important crops like soybean, corn, and wheat remains highly recalcitrant to Agrobacterium mediated transformation. So how this problem being addressed? How we can solve this?
So when we say transformation, different laboratories have different definitions of what transformation is. The majority of laboratories look at transformation as a process of getting DNA into the plant cell and regenerating that plant cell back to a whole plant. So this is both transformation and regeneration. Our laboratory concentrates on the transformation process itself. So for many plants, especially monocot grasses, the block point is in regeneration. And so you can get DNA into the cells, but it's hard to regenerate entire plants back from these cells. This has been aided recently by using certain developmental or morphogenic genes that people have put in to help the regeneration process. But on top of that, or prior to that, we know that the actual transformation process itself can be blocked. For example, we know that in rice and in Arabidopsis, there are many genotypes or ecotypes that are very, very difficult to transform, even before you try to regenerate. And so there's a wide variety of susceptibility and resistance to just the transformation process itself. And we're trying to address this by understanding what the plant genes are that are involved in the transformation process, and then trying to manipulate these genes so that we could improve transformation. Then people, if they wish, could also use these developmental or morphogenic genes to look at regeneration. But there are two basic problems here. It's getting the DNA in and then regenerating the cells.
Okay, that's great to know. So you mentioned that you are able to manipulate some genes or proteins to improve the Agrobacterium transformation efficiency. So could you please explain some of the genes and proteins we can use them?
So many, many years ago, we identified more than 100 what we call Arabidopsis rat mutants. Rat means resistant to Agrobacterium transformation. And we have more than 100 of these. As far as we know, none of them are duplicated yet. And so there are at least 100 different genes somehow involved in transformation. And when I talk about transformation with Arabidopsis, I talk about root transformation, somatic cell transformation, not flower dip, because we've shown that flower dip transformation obeys very different rules from somatic cell transformation. And when people are trying to transform and regenerate crop plants, they are generally doing it with somatic tissue, not with germline tissue. So we have only identified a handful of these genes through these rat mutants and through other methods. And the one that we've had the most success with and the most characterization are various histone genes, plant histone genes. So Kieran Mysore, when he was a graduate student in my laboratory, showed that a particular histone H2A gene, when mutated in Arabidopsis, severely decreased tDNA integration, although it didn't really decrease to a really large extent tDNA transfer. So that suggested that this histone H2A gene is involved in integration. However, when Gabriela Tineo was a postdoc in my lab, she and other people showed that if you overexpressed certain histone genes, but not all histone genes, you could increase transformation, but sometimes this would also increase transient transformation as well as stable transformation. And the group then went on to show that this probably has a protective effect on the DNA as it's coming into the cell. So we do know that a lot of histone genes are involved in transformation. We don't know exactly how they're involved in transformation. It's something that we're starting to look at right now.
Okay, great. So are you using any like morphogenic genes? Did you check apart from that your genes, like what you are working?
We have not, at least in our rat mutants collection, we have not uncovered any morphogenic genes. And we probably wouldn't, because our assays are infecting roots of axenically grown Arabidopsis plants, and we're not trying to regenerate plants. We're just growing callus from root tissue. So that's how we sort of separate out the transformation and the regeneration. We look at transformation for just the ability to get DNA in and express either transiently or stably into callus tissue without having to regenerate. So we've unlinked the regeneration and the transformation.
Okay, which one do you feel like more successful, like a transient or a stable one with your genes?
With the histone genes?
Yeah, with histone.
Well, overexpression of certain histone genes, it's not all histone genes, but certain histone genes will increase both transient and stable transformation. So, and more recently, Kieran Mysore has shown that if you put a type 3 secretion system into Agrobacterium and put a type 3 secretion signal onto histone H2A, it could bring histone H2A into a plant and increase both transient and stable transformation. So this is equivalent to what we did by overexpressing a transgene in the plant, but instead it's actually just bringing a protein into the plant, which is a lot easier to do. So our laboratories are working together to improve this process right now.
Great. Yeah. Hopes to see the new results and new genes which can use for increasing the transformation efficiency.
So I should say that it's also known that plants put up defense responses against Agrobacterium, and these defense responses may be involved in inhibiting transformation. And what Kieran has showed is that if you take certain AVR genes, effector genes from Pseudomonas, and transfer those proteins from Agrobacterium to plants, you can increase transformation by decreasing defense responses. So there are a lot of different genes probably that are going to be able to be used to increase transformation.
So do you have any specific like strains like you feel those are more successful with your transformations?
Well,
there are so many strains.
yes. Well, there's so many strains, but there are actually very few strains that people use. So there are commonly used strains for plant transformation, disarm strains. EHA 101, EHA 105, LBA 4404, GV 3101, AGL 0, AGL 1, and some rhizogeny strains. And that's pretty much it for what most people are using. But there are hundreds or thousands of strains out there that are starting to be characterized now. And people are starting to look at these strains and say, are any of these strains, for example, more virulent on certain plant species? The other thing is that certain strains seem to be better suited for certain plants. For example, the BO542 TI plasmid background, EHA 105 and AGL1, for example, those strains are extremely useful in solanaceous plants. They are less useful, but still useful, but less useful, for example, in Arabidopsis, whereas in Arabidopsis, GV3101 is by far the most virulent of the strains that have been tested. So it's a matter of matching strains with the hosts. And even so, for example, in Arabidopsis, there are some Arabidopsis ecotypes that are extremely recalcitrant to root transformation, even by GV3101, which is the standard strain that's very successful, used for many ecotypes such as Columbia and various other ecotypes, WS, things like that.
Okay, great. So you
So I'd
have
just like to say that one of the important things here is going to be developing new Agrobacterium strains, disarming other Agrobacterium strains, and developing them for use, and perhaps we'll find strains that are better suited for certain crops.
Yeah, that's correct. Yeah. And I agree with you. So you have been many years working with Agrobacterium. So what motivated you to pursue a career in this field?
Okay, so I've been working with Agrobacterium now for, I think, 46 years.
Wow.
And yeah, it's been a long time. And so when I was a graduate student at UC San Diego in Steve Howell's lab, I heard a seminar. And in this seminar, this was sort of pre-Agrobacterium days, or the verge of Agrobacterium. It was in the mid-1970s. And somebody gave a seminar about protoplast transformation with naked DNA. But somewhere in that seminar, the person said, watch for Agrobacterium. People are thinking that perhaps, although it hadn't been shown at the time, Agrobacterium could transfer DNA. And watch for this as something that may develop in the future. And that one little line in the seminar, and the seminar wasn't even about Agrobacterium, sort of piqued my interest. So I went to the library and read everything that I could about Agrobacterium. It wasn't the huge literature, the volume that there is now. And I decided that this is what I want to work with. And I contacted Gene Nestor and Mary Del Chilton in Seattle, asked them whether I could do a postdoc in their laboratory after I had finished my PhD. And they said yes. And that's how I got into it.
So you work with the postdoc Agrobacterium, and then you became a professor.
Right. My graduate work was on Chlamydomonas chloroplast genes.
Oh, okay.
So it was a bit of a shift, but it's always good to do different things.
Yeah, that's correct. Yeah. So do you have any advice for the next generation of plant scientists?
Yeah, and I've been thinking about this actually. And this is just my advice, but it's not necessarily unique to me. And there's 4 things I would like to suggest people do. Number 1, choose a really good question. Don't choose a species, don't choose a technique. Choose a really good question that you want to answer, then find the appropriate species and the appropriate techniques to answer them. A lot of people do what I call Me Too biology. Somebody's done this in tomato, so I'm going to see if the same thing happens in potato. You'll get some publications out of it, but you're really not advancing our knowledge of how the natural world works. So asking a good question is the key first thing to do. Secondly, train, this is my particular advice, train as a basic scientist. Even if you want to do applied research, and that's fine. People from my laboratory have gone into basic science, they've gone into applied research. Many of them have left, most of them have left Agrobacterium, and many of them have even left plant biology and have gone into human and animal pathogenesis. All that is great. But I think it's really important to understand how science works first. And then if you want to apply it to something, that's great. But a fundamental understanding of how nature works, I think, is really important. Linked to that is, I think it's very important to read very broadly, not just in your own area, and to go to all sorts of seminars, things completely unlinked for what you're doing. You may want to go to cancer seminars or neuroscience seminars or whatever, because you never know what sort of ideas you may get from a different way of thinking and a different system that you could later apply to what you're doing yourself. And the last thing is what my advisor, Steve Howell, told me, and I think it's the best advice, he gave me, and I really highly recommend it. When doing a postdoc, do something completely different from what you're doing as a PhD. I was working on Chlamydomonas chloroplast DNA. Biology department, I went to a microbiology department and learned about Agrobacterium as a microbial genetic system as well as on plants. And the experience of doing different things is really going to help you in the future. If you want to come back to your PhD work, that's fine. You know, you can always do that later. But by having a broad knowledge of many different types of things, different approaches, different systems, it will make you, I think, a better scientist. I think it'll make you more marketable also. And let's face it, what you're doing today, 10 years from now, may no longer be sexy, which means it may no longer be fundable. If you only know how to do one thing, you're going to be in trouble. But if you know how to do different things, and I'm not just talking about techniques, I'm talking about ways of thinking about things in different systems, that's really going to help you adapt to the developing scientific situation as it occurs during your career. So get lots of different experience.
Yeah, I agree with you. It's really a matter good thing to know many things, and also you can think very broadly about the science. So staying in the same
This
field.
is the advice that I've given to my graduate students, and nobody went off to do a postdoc in Agrobacterium, although some of them did come back to work on Agrobacterium afterwards. And having this extra experience and extra knowledge and different ways of thinking about things, I think, help them all in their future work.
Yeah, you are right. But so how when you apply the positions, like if they mostly like to take the same experience people, like when so how to avoid that problem, like so you have one
Well, I
field
don't
of
know. It certainly is
uh,
an issue for getting a postdoc. I don't know how much of an issue it is getting for a job. So, for example, postdoc advisors, when they're looking for postdocs, really would like to see people who are trained in their particular area, because you can come into the lab, plug into the lab immediately, and start producing data. Okay. And from the graduate student's point of view, yes, if you know how to do something as a PhD student and you go to a postdoc lab and do the same thing, absolutely, you will get more publications out more quickly. You're not necessarily learning completely new things. So as I said, later on in your career, you may be stumped because you just don't know how to do a broad range of things. So I think it's important to get this broad range of experience. You may, it may take you longer to get your first papers out of your postdoc lab, but it will help you, I think, tremendously in your eventual career. And I think that's very important. When people go for jobs, usually they're interviewing on the basis of their postdoctoral work. And so if you do really good postdoctoral work, that's one of the ways to get a job backed up by your graduate work. So, you know, you're going to spend an extra year as a postdoc. Okay, you get your first paper.
yeah.
Okay,
Okay.
everybody's going to have to get papers out really fast. Okay, and your postdoc advisor, of course, wants papers out as fast as you can to help his laboratory. But look at it this way: if you have another 40 years of research experience ahead of you, that one extra year really doesn't mean that much in terms of total time, but it can mean a huge amount of difference in terms of experience and how that can affect you later on for many different things. So.
Thank you so much, Stan, for joining me today. It was nice talking with you.
PlantGENE would like to thank the National Science Foundation for their support. To learn more, please visit their website at nsf.gov.